Assalamu alaikum dear students, welcome to your channel the Anatomy Canvas, and in today's class we are going to discuss with you the development of the male and the female reproductive system. So the learning objective for today's class are that we will discuss about the development of the gonads, that is the primitive sex glands. We will see the development of the genital ducts and the external genitalia. So development of the genital system. The genetic sex is established at the time of fertilization by the kind of the sperm that fertilizes the ovum. So key to sexual dimorphism is the Y chromosome, which contains the SRY gene on the Y chromosome. The gonad begin to attain the sexual characteristics from the 7th week. Early the genital system in two sexes are similar, and this initial period is called the indifferent state of the sexual development, or the ambisexual stage. So main important factor is the Y chromosome, which is key to sexual dimorphism, and it carries the SRY gene, that is the sex determining region on the Y chromosome. And before 7th week there is the stage in which both there is no difference between the different sexes, that is called the indifferent state or the ambisexual stage. So, chromosomal and genetic sex is established at time of fertilization. The type of the gonad that develop is determined by the sex chromosome complex of the embryo, either XX or XY. Before 7th week, of gonads of two sexes are identical, that is indifferent or ambisexual stage. Male phenotype requires Y chromosome, the SRY, that is the sex determining region, and Y gene for our testis determining factor. The female phenotype requires two X chromosome with number of the genes. So key to sexual dimorphism is Y chromosome, which is SRY gene. Absence of Y chromosomes leads to development of the ovaries as the default mechanism. So please remember this very important MCQ in this slide is that before 7th week there is indifferent gonad stage of indifferent or identical gonads or ambisexual stage. First important MCQ, and second important MCQ is that the Y chromosomes carry the SRY gene, which releases the factor that is called the testis determining factor, which secretes the testosterone and which is responsible for formation of the testes. And in absence of the Y chromosome, the ovaries develop as a default mechanism. So these are all your MCQs. Development of the gonads, that is testes and ovaries. The main very important point is that essentially 4 different cell lineages contribute to the gonads. So this is another MCQ as well, that the 4 different cell lineages they are involved to form the gonads. The first are the proliferating coelomic epithelium on medial side of the mesonephros. Second is the underlying mesonephric mesenchyme. So the mesonephric mesenchyme is the mesenchyme which is present in the region of the mesonephros. Then invading angiogenic mesenchyme, which is already present in the mesonephros. So as mesonephros is present and it is forming the primitive kidneys, so angiogenic mesenchyme is also present, and it is also part of the one of the cell lineage which is responsible to form the gonad. And the fourth very important, that is the primordial germ cells. So another very important MCQ over here is that they arises from the epiblast very early in development, and then they migrate from there to the allantoic wall, where they are present and where they stay for some time, and from that area then they migrate through the wall of the yolk sac, from the wall of the yolk sac through the dorsal mesentery into the area of the genital ridge. So remember, 4 cell lineages are involved in the formation of the gonads: coelomic epithelium, mesonephric mesenchyme, angiogenic mesenchyme, and primordial germ cells. So in this diagram, you can see over here that this is the area of the mesonephros which is developing, and behind this mesonephros you can see the mesonephric duct is present, and in this location you can appreciate the metanephric mass and metanephric duct is present, which is entering into the urinary bladder. Here you can see this is the yolk sac, and from the wall of the yolk sac these are the primordial germ cells which are migrating by means of the amoeboid movement to the area of the genital ridges. So in this cross section you can see this is the wall of the yolk sac and this is the area of the dorsal mesentery, and from here the cells are migrating, the primordial germ cells which migrate to the areas of the genital ridge. Here in this area you can see this one is the paramesonephric duct, and this is the mesonephric duct along with mesonephros. So paramesonephric duct is present lateral to the mesonephric duct. This one is the developing cortex and medulla of the adrenal gland. Here you can see the aorta which is forming, and again on this side you can see the genital or the gonadal ridges. So after the arrival of the primordial germ cells, you can appreciate over here that these primordial germ cells then they activate the gonadal sex cards in the cortex, and here in this diagram you can see that the sex cards are developing in the area of the cortex. So in this diagram you can see, you can understand very well that how these cells migrate. Initially they are derived from the epiblast, and then they migrate from the epiblast, and then they stay in the wall of the yolk sac, and later on they migrate from the yolk sac through the dorsal mesentery into the area of the genital ridges. So these primordial germ cells, these are the large spherical sex cells. They are visible early in 4th week among the endodermal cells of the yolk sac near the origin of the allantois. During folding of the embryo, dorsal part of yolk sac is incorporated into the embryo. Thus, primitive germ cells migrate along the dorsal mesentery of the hindgut to the gonadal ridges. So in this diagram also you can see that this is the area of the gonadal ridge. Here you can see this is the area of the dorsal mesentery, and you can see the migrating primordial germ cells which are migrating through the dorsal mesentery into the area of the genital ridges. So during 6th week the primordial germ cells enter the underlying mesenchyme and are incorporated into gonadal cords. Here you can see in this area their incorporation into the gonadal cords. So again in this diagram you can see, you can appreciate that this is the area of the yolk sac. Here you can see this is the hindgut, this one is the mesentery of the hindgut, and the cells they are moving through the hindgut and its mesentery into the area of the genital ridges. These are the genital or gonadal ridges. This one is the mesonephric duct, and behind and lateral to it is the paramesonephric duct. Then in this area you can see also that after their arrival they are migrating through the dorsal mesentery into the area of the genital ridge, and then they are incorporated and they are forming the primary sex card. Then the fate of the primary sex card is then different in both the sexes, in both male and female, and we will discuss it in detail in the next slides. So first we will study the indifferent gonads. The formation of the gonad is first indicated by appearance of an area of thickened coelomic epithelium on medial side of mesonephric ridge in 5th week. Here in this slide you can see this dark blue color area. This one is the mesonephros, and this area, this swelling over here which is present in this area, this is the developing gonad which is appearing on the medial side of the mesonephric ridge. The more and more cells are added to this area, the epithelium, coelomic epithelium proliferate over here, and this gonadal ring continues to grow grow in a longitudinal direction till and proliferate till it reaches the caudal end, and also because of the proliferation it displays the renal corpuscle of the mesonephros in a dorsolateral direction and form projection into the coelomic epithelium, and this projection is called the gonadal ridge. So this one was the mesonephros, and in front of the mesonephros on its medial side you can appreciate this area. This is the gonadal ridge. So because of this gonadal ridge, when it is developing and it grows longitudinally and it reaches up to the tip of the mesonephros, so it divides this area, it divides mesonephric ridge into two parts. The one which is a lateral part is called the tubal fold. It contains the mesonephric and paramesonephric ducts, and the one which is medial part is called the gonadal ridge. So this darker area is the medial part, and behind this the lateral part, which is tubal part, which contains both the mesonephric as well as the paramesonephric ducts. The same slide at high magnification you can see. So this one was the mesonephric duct, and over here this is the gonadal ridge which is developing. This one is the developing GIT, that is the midgut, the foregut and the hindgut, and here in this area you can see the metanephros which will form the kidneys. At this level the section is taken, and you can see over here this one is the midgut. As you can appreciate over here, this is the midgut. So here you can also see this is the area of the midgut. This whole is the genital ridge where the gonads are developing, and this part is the medial part of the genital ridge, the mesonephric ridge where the gonads are, the genital ridge is forming, and this one is the lateral part which is the tubal part where you can see both the mesonephric and the paramesonephric ducts are developing. So this one is the lateral part of the mesonephric ridge, and this is the medial part. Here you can see the somite which is present, the neural fold along with the notochord you can also appreciate, and this one is the amniotic cavity. Now the almost a similar diagram in a longitudinal section. You can see that these are the intestinal loop, and this one is the midgut through the yolk sac, and this hole is the mesentery of the midgut. And here you can see this one is the mesonephros, and it's a duct which is present behind, and on its medial aspect you can appreciate the gonads which are present, the genital ridge which is forming. So now we will see how these primordial germ cells, when they arises in the region of the genital region, they form the testes or the ovaries. So finger-like epithelial cards, these are called gonadal cards, grow into underlying mesenchyme. Here you can see this one is the coelomic epithelium, and this coelomic epithelium continues to proliferate and increases in its stratification, its number of layers, and then it forms the primitive gonadal cards or primitive sex cards over here. So at in different stage, the primitive sex cards are present in the cortex, and they also move in the medulla where they displace the developing mesonephric area, the mesonephric nephrons and the glomerulus to the lateral side, and they also reach up to the level of the medulla. So this one was the indifferent stage when there is no difference, and here you can see the primordial germ cells which are arising over here. What happens next? If the embryo is XY, the medulla differentiates into testes and cortex regresses except for the vestigial remnants. So in this diagram, this one is taken from the male. Here you can see the cortical cards which initially starts in the cortical area, the primitive sex card. They migrate and they move into the area of the medulla, while in the area of the cortex they almost disappear and they regresses. So development of the gonads and associated ducts to show the fate of the primordial germ cells. So here you can see the primordial germ cell. This one is the female. The fate of the primary sex card is different both in the male and the female. So in case of the males, when the testis is going to develop, the testis determining factor induces the gonadal cards. These are the seminiferous cards to condense and extends into the medulla of the indifferent gonad. So here you can see the primitive sex cards which move towards the medulla, and they form the medullary cards as well, where they anastomose over here and they form the rete testes. The dense layer of the connective tissue which is present over here, which separates these primitive sex card from the surface, and this is called the tunica albuginea. In 4th month, the testis cards become horseshoe shaped and their extremities are continuous with those of the rete testes. So here you can see in this diagram also that the rete testes are present. Over here the cortical cards almost disappear, they regresses, and the medullary cards develop, and these medullary cards form the seminiferous tubules. They make a connection with the rete testes, and in turn rete testes they form the efferent duct. They are attached to the efferent ductules which are formed from the mesonephric ducts, and then they are attached to the epididymis. So in males, you can also appreciate in these slides that interstitial cells of the Leydig derived from original mesenchyme of gonadal ridge begin develop shortly after onset of differentiation of these cards. So here you can see this one, the coelomic epithelium, and this coelomic epithelium, when they were forming the primary sex card, they encloses the primordial germ cells. Their connection from the surface is lost. Then the primitive sex cards also disappear and medullary cards are present. The coelomic epithelial cells originally which were present over there, they form the Sertoli cell. So another very important point to remember over here is that these Sertoli cells continue to proliferate throughout the prenatal life and also in very early childhood. But when they stops to proliferate and they maturate, they cannot be reactivated. This is very important point, that the Sertoli cells, once they mature, they cannot be reactivated. And another important point regarding Sertoli cells which are present over here is that the Sertoli cells can support a fixed number of the germ cells population. So this is very important because it determines the maximal limit of the sperm output. So these Sertoli cells are derived from the coelomic epithelium. The cells which do not enter into the sex card remains over here in this area, and also the cells which are present in the mesonephric mesenchyme, they will form the Leydig cells. So these Leydig cells lie between the testis card and they begins production of the testosterone by 8th week. Thus, testis is able to influence the sexual differentiation of the genital ducts and the external genitalia. This is also very important MCQs, that the testosterone which is secreted by the Leydig cells, these Leydig cells originate from the mesonephric mesenchyme and also small amount from the coelomic epithelium as well. These secrete testosterone in 8th week, and after the secretion of the testosterone, differentiation into testes starts. So the testes cards remain solid until puberty. When they are canalized, they form the seminiferous tubules. Thus they join the rete testis tubule, which in turn enter the ductuli efferents or efferent ductules, which are the remaining parts of the excretory tubules of the mesonephric system. They link the rete testis and mesonephric or Wolffian duct, which becomes the ductus deferens. So development of the ovaries next, very important, that the gonadal development occurs slowly in the female. It is little behind that of the male. In X embryo, primitive sex card dissociate into irregular cell clusters. So in this you can see the first that the primitive sex card they dissociate, then a secondary generation of the sex cards appear, and these sex cards then they surround the primary oocyte and they form the primordial germ cells. The original coelomic epithelial cells form the follicular cells which supports the primordial germ cells, and in this way primordial germ cells and primary and primordial follicles and primary follicles are being formed. So in this you can see the surface epithelium of female gonads, unlike that of the male, continue to proliferate, giving rise to a second generation of the cortical card in the 7th week. So this one was the second generation of the cortical cards. These cortical cards penetrate underlying mesenchyme, but they remain close to the surface. So here you can see this is the area where these cortical cards are present. They are present close to the surface, and they surround the primordial germ cells, the oogonia, and they form the primordial follicles are the primary follicles, and the cortical cards which surround this oogonia, they form the follicular cell. So in the medullary region you can see the mesonephros and mesonephric mesenchyme and angiogenic mesenchyme which is present over here. It will form the medulla of the ovaries, and the remnants of the mesonephric system is the rete ovaries which is present over here. A connective tissue is present in this area which separates these primordial germ cells from the surface epithelium, and this connective tissue is called the tunica albuginea. So now we will see the differentiation of the genital duct system first in indifferent stage. So in this you can see these are the developing mesonephros along with their duct which is called the mesonephric duct. We will see the detailed development of the mesonephric system with the development of the urinary system. Here you can see this one is the mesonephric duct, and lateral to this mesonephric duct there is invagination of the coelomic epithelium, which initially is present in the form of the ridge. It forms a tubular structure, and it forms the paramesonephric duct. This paramesonephric duct in its starts till the lower part of the mesonephric duct is present lateral to it, then it crosses it anteriorly and it comes to lie medial to the mesonephric duct. So here in this slide you can also appreciate that this one was the mesonephric duct and the mesonephric tubules and mesonephric system which is forming mesonephros, and this is the paramesonephric duct which is again forming from the invagination of the coelomic epithelium. So again you can see this paramesonephric duct has three parts. The first part, the second part and the third part. The first and third part are vertical and second part is horizontal. First part is present lateral to mesonephric duct. Second part crosses the mesonephric duct from the lateral to medial side anteriorly, and then to the two paramesonephric ducts of the sides come closer to each other. They fuses with each other, and then they enter into the urogenital sinus. So this one is the development. Then the developmental, there are the different changes both in case of the males and the females. So we see how these occur. In this slide you can also appreciate that this was the yolk sac, and here you can see the allantois, the GIT which is forming. Here you can see the urogenital sinus which is present, and in this area you can see the mesonephric duct which is paramesonephric duct in pink color. You can appreciate it over here. In blue color you can see the mesonephric ducts which are present over here, and these two ducts moves downward, while here you can see this is the metanephros which is forming the kidney, and these move upward. So because of the positional changes of both metanephros and mesonephric and paramesonephric ducts, the paramesonephric duct comes to lie closer to each other. They fuse it and they enter into the sinovaginal tubercular urogenital sinus. So again in this slide, this one the slide in indifferent stage in which both the mesonephric duct and the paramesonephric both are present. This is the metanephros which is forming the kidney, and this is the metanephric tubule which will form the ureter. This is one is the developing urinary bladder, and here you can see the urethus, and here this whole is the urogenital sinus which is separated by the urorectal septum into urinary part and the rectal part. Then the differentiation between the two male and the female is different, and we will see in that in case of the male There is the development of the, here you can see, start from here, this one was the testes and the mesonephric duct, and mesonephros continues to develop. So mesonephros will form over here the efferent ductule, the appendix epididymis and paradidymis, while the mesonephric duct will form the epididymis, then the vas deferens, until the point of the ejaculatory duct. So this was the whole the development of the mesonephric system, and it is occurring under the effect of the SRY gene, which is present on Y chromosome, which activate the Leydig cells to produce the testosterone, and which will form the formation of the testes and development of the mesonephric duct. While the paramesonephric duct system in males almost disappear completely except for a small part which is called the appendix testes, which is present over here, and the prostatic utricle. In case of the female, the paramesonephric system continues to develop. So two paramesonephric ducts, as I have told you, that these paramesonephric ducts have three parts: first vertical, second horizontal, and third vertical. The third particle fuses with each other to form the uterus, and first and second part will form the uterine tubes, while the mesonephric ducts in these cases disappear, and mesonephros as such will form the epiphrone and the paraophrone, which is present in this area, in the area of the ovary. Epiphrone and paraophrone both are formed from the mesonephros, while the mesonephric duct will form the Gartner duct. So these are all very important MCQs. The remnant of the mesonephric duct in the female system and the remnants of the paramesonephric ducts in the male system, they are all very important MCQs. So here in the table you can see the gonads, the testes in males and ovaries in female. The gubernaculum forms the gubernaculum testes in male and ovarian and round ligament. The mesonephros, this is the body of the mesonephros, form appendix epididymis, efferent ductules, lobules of epididymis, paradidymis, and aberrant ductules, while in case of the female, appendix vasculosi, epiphrone, and paraphrone. The mesonephric duct in the males form duct of epididymis, the vas deferens, the ejaculatory duct, and part of the bladder and the prostatic urethra. While in case of the females, the mesonephric duct will form the duct of the epiphrone, that is Gartner duct, and part of the bladder and urethra. Each and every line in this table is MCQ, so please memorize this table for your paper. Also, the paramesonephric ducts form appendix of the testes in the males and the prostatic utricle. These are remnants of the paramesonephric ducts in males. While in case of the males, they form uterine tube, uterus, and part of the vagina. The allantoic duct form urachus in both the sexes, and cloaca, its dorsal part, will form the rectum and upper part of anal canal in both the sexes. Ventral part form most of the bladder and both the sexes, in urethra also in the female, and part of the prostatic urethra is from ventral part, while urogenital sinus will form the prostatic urethra distal to utricle, the bulbo-urethral glands, and rest of urethra to the glands. And in case of the female, the urogenital sinus will form the greater vestibular glands and the vestibules. Genital folds will form the ventral penis, and in females form the labia minora. Genital tubercle form penis, and the urethra in the glands, while in female forms the clitoris. So again, this table is also very important for MCQs, so please also memorize this table as well. Almost a revision, the testes will form the testosterone from the Leydig cells, and Sertoli cell will secrete the Mullerian inhibitory substance, which will inhibit the paramesonephric ducts, while the Leydig cell, they stimulates the mesonephric ducts. They also secrete the dihydrotestosterone, which will stimulate the formation of the external genitalia. Similarly, ovaries, in the absence of these two, ovaries will form, and ovaries will secrete the estrogen and the placenta, the estrogen, and these estrogen will form the development of the paramesonephric system and the external genitalia. Again, almost the same, the genital duct system in the males. That is the this one was the mesonephros and mesonephric duct, which is forming over here the remnants. The caudal part of the mesonephros will form the paragenital tubules, and its cranial part have connection with the rete testis and form the epigenital tubules. These again the almost the same slide. The mesonephric ducts persist and form main genital system except for most cranial part, the appendix epididymis. So the mesonephric duct elongate, highly convoluted, forming the ductus epididymis. Immediately below the entrance of the efferent ductules. The mesonephric ducts obtain a thick muscular coat, the ductus deferens, from tail of epididymis to the outbutting of the seminal vesicle. And also remember, this muscular coat rises from the mesenchyme, that is the splanchnic mesoderm of this area. The region of the duct beyond the seminal vesicle is ejaculatory duct. Paramesonephric duct degenerates except for a small portion, which is the appendix testis. And again, this point very important, the appendix epididymis, it is formed from the mesonephros itself, not from the duct. So female duct system, if testis fails to develop a gonadal male, similar development of mesonephric duct occurs in male because of the absence of the Mullerian inhibitory substance. Development of the male genital glands, again another important, the lateral outgrowth from the caudal end of each mesonephric duct give rise to the seminal vesicle or the seminal gland. So here you can see this is the ejaculatory duct, and these are the seminal vesicles which are forming from the caudal part of the each mesonephric ducts. Multiple endodermal outgrowth arising from prostatic part of urethra grow into surrounding mesoderm, and they will form the prostatic glandular epithelium. So here you can see these are the prostatic glandular epithelium which are forming. The bulbourethral glands develop from the paired outgrowth from the spongy part of the urethra. These are all also very important MCQs, because bulbourethral gland develop from the spongy urethra, and the prostatic urethra, the prostatic glandular epithelium differentiate into prostatic urethra. Similarly, in females, as I have told you, the paramesonephric duct, the first and second part will form the uterine tube, and the third part fuses with each other to form the uterus. Then you can see the upper part of the vagina, which is also formed from the paramesonephric duct as well, and this vagina has a dual origin. Its upper part formed from the paramesonephric duct, while its lower part formed from the sinovaginal bulbs. So genital ducts in female, second part of paramesonephric duct fuses, they move mediocaudally, and urogenital ridges gradually comes to lie in a transverse plane. Because of these positional changes, the transverse pelvic fold is raised, which is forming the broad ligament of the uterus. This broad ligament of the uterus and uterus itself separate the pelvic cavity. So the anterior cavity between the urinary bladder and the ureter is uterovaginal pouch, and the cavity between the uterus and the rectum is the uterorectal pouch. So these are the two different cavities which are present in the females. Early development of the uterus and the ovaries. So again, this is almost the same diagram. Here you can see the developing paramesonephric ducts as well. So very important is then the development of the vagina. So as I have told you that the vagina have dual origin. When the paramesonephric ducts touches the area of the urogenital sinus, the two solid folds appear, and they grow towards the developing paramesonephric ducts, and they come to lie on each side of the paramesonephric ducts. So when canalization in this area occur, they will form the fornices. The junction of the paramesonephric duct and that of the sinovaginal bulb is present at the level of the hymen, which is a thin plate of the tissue and which is usually perforated in the females. So by the 5th month, vaginal outgrowth is entirely canalized and vaginal fornices will form. Then development, it has two origin or dual origin, upper part from uterine canal and lower part from the urogenital sinus. Lumen of vagina remains separated from that of the urogenital sinus by thin plate of the tissue, the hymen. As I have told you, at the junction of these two, its origin at the junction point between the cavity of the vagina and urogenital sinus, there is thin plate of the tissue, the hymen, which separates the two cavities of two from each other. So remnants, as I have told you, the remnants of the ducts in the females, the epiphrone and the paraephrone, these are the remnants of the mesonephros, and the Gardner duct is the remnant of the mesonephric duct. Congenital anomalies, these are very important. Also, the double uterus or uterus didelphys result from failure of the fusion of the inferior part of the paramesonephric duct. So, it may be associated with double or single vagina. Bicornuate uterus, in which one paramesonephric is retarded in its growth and does not fuses with the other one. Bicornuate uterus with rudimentary horn, in which rudimentary horn is present and may not communicate with the uterine cavity. Unicornuate uterus, in which one paramesonephric duct fails to develop, resulting in the uterus with one uterine cavity, one uterine tube. Absence of vagina and uterus, it can occur, results from failure of the sinovaginal bulbs to develop. Vaginal atresia can occur because of failure of the canalization of vaginal plate, and imperforate hymen can occur, failure of the perforation of inferior end of the vaginal plate. So here in this slide you can see this one was the normal uterus. In this case you can see the double uterus, the separate paramesonephric duct forming the bicornuate uterus. In this you can see the double uterus with a single vagina. In this case two vagina are present, and in this case single vagina is present. In this case you can see the bicornuate uterus. Two separate cornu of the uterine cavity with the rudimentary horn are present, and in this case you can see bicornuate with rudimentary horn. In this you can see the septate. So in the center a septa is present, which separates the cavity of the uterus into two parts, and in which one side do not develop or regresses, and on other side the paramesonephric duct forms the unicornuate uterus. So I complete here my today's class, and we will do inshallah the external genitalia in the next class. The references for the today's class are the Lyman's Medical Embryology, The Developing Human by Keith Elmore, and Gray's Anatomy by Susan Stranding, the 40th edition. Thank you very much for your attention for today's class, and Allah Hafiz till the next class.